Let's continue with our discussion of cells. This is many. Let's talk about the things that are common to all cells. The plasma membrane defines the cell. The plasma membrane is a membrane, very thin membrane, around the outside of the cell. There may be a cell wall in addition. If there is, the cell wall is going to be outside of the cell membrane or plasma membrane. We can use either term. The cytoplasm is inside of the plasma membrane. Cytoplasm is the stuff that's inside the plasma membrane, and in the case of eukaryotic cells, it will be the stuff between the nuclear membrane and the plasma membrane. The cytoplasm is made of cytosol, which is the fluid part or the aqueous part of the cytoplasm, and then there are organelles as well. So we'll talk about the organelles. Those are structures made of lipids and proteins inside the cell, and they have particular functions. DNA is in all cells. DNA is the genetic material of the cell. That's a polymer, deoxyribonucleic acid, and it's made of nucleotides. Its function is to store genetic information. All cells have ribosomes, which synthesize proteins. So ribosomes are going to use the information provided by DNA to synthesize proteins. Synthesize means to make. All right. So this is just a diagram. We talked about things that are in all cells. Now let's break it down a little more. Prokaryotes. Prokaryotes are going to include bacteria. They do not have a nucleus. Their cells do not have a nucleus, and they do not have other membrane-bound organelles. They have the plasma membrane. They generally have a cell wall, but they don't have a nucleus. That DNA is just off on its own out in the cytoplasm. Most have a cell wall. We believe that they are very much like the first cells, and it includes two domains, both the bacteria and the archaea. We will not really discuss the archaea in human biology. They are organisms that are found in extreme environments, places like the hot springs in Yellowstone National Park, places that are extremely salty, halophytes, and things that have an extreme osmotic pressure, like some things that are very sugary. I've found them on maple syrup. And then the bacteria are prokaryotes. And those are two different domains. Those are considered two different domains, the archaea and the bacteria. And then the eukarya are another domain. And the eukarya includes several kingdoms. We will talk about bacteria only a little bit, mostly in terms of that they can be pathogens. Not all bacteria are pathogens. Many bacteria are beneficial. We have beneficial bacteria living in our intestines. We have beneficial bacteria associated with crop plants. But some are pathogens, and so we're aware of that in public health and in human biology. All right, so let's just talk generally about prokaryotic cells. In human biology, we're not going to discuss them at length. That chromosomal DNA is in something called a nucleoid. So see that ending there, that O-I-D ending of that word means in the shape of or something similar to. So that chromosomal DNA is kind of bunched up together, and we call it a nucleoid, as in something similar to a nucleus. It does have ribosomes in the cytoplasm. It's got ribosomes loose in the cytoplasm. Right. We've got the cell membrane. Right. We've got the cell membrane surrounding the cytoplasm, and then the cell wall is outside of the cell membrane. We've got a flagellum on this one. This is just a generalized idea of a bacteria. The ribosomes are just little dots in here. There's our membrane. There's our wall outside the membrane. In this case, we also have a capsule that is often a polysaccharide, meaning it's a slimy, sugary mess that sometimes protects the bacteria from the immune system. And then we've got some little pili there that can allow for exchange of genetic material. All right, let's keep going. Prokaryotic cells are smaller than eukaryotic cells. They're smaller and they're less complex. So this is favorable to moving material in and out of the cells, right? And we don't have any internal movement of things inside the cell. So when we talk about the eukaryotic cells in a minute, we'll see all those organelles that allow for substances and molecules to be moved around. So prokaryotic cells can't really do that for themselves, so they just need to be smaller and let diffusion and osmosis take care of that for them. Let's think for a little while about why cells are small, right? Cells are small, and that's because of our surface area to volume ratio. So as a cell increases in size, the volume of that cell gets bigger faster than the surface area does. So imagine that these three cubes are representing cells of different sizes. Here we've got a small cell, and its dimensions are a unit of 1 in each direction. And we have a total volume of 1 and a surface area of 6. As we increase to this larger size, we've got a dimension of 3 in each direction. And so we have a total volume of 27, and the surface has increased to 54, but our ratio is smaller. Our surface to volume ratio is 2, where with the smaller cell, our surface to volume ratio was 6. And we can also imagine that if there's some stuff inside that cube, it's now farther away from the surface, right? In this cube, the farthest anything is from the surface is 1/2 of a unit. Here, it can be a unit and a half from an outside surface. One way to think about surface area is to imagine a child's sports team, and now it's the end of the game, and the players are going to shake hands. Instead of sending one player from each side out to shake hands and send them back, you line up both teams and they walk along, slide past one another, shaking hands. That's an example of surface area. So pay attention to surface area concept as you move through the body systems. Let's consider eukaryotic cells. Eukaryotic cells have a nucleus. Plant cells, animal cells, and fungal cells are eukaryotic cells. And we have a nucleus. It's often shown in purple, but it is not purple. These colors in this illustration are just to differentiate the different organelles in the cell. We'll hear that word organelle. That means little organs. So these are structures inside the cell that have functions. True organelles have membranes, and we call those membranous organelles. So if we say membranous organelle, it has a membrane and is considered a true organelle. But we'll also discuss ribosomes. Ribosomes do not have a membrane, but we consider them an organelle for the purposes of this discussion. All right, there's our nucleus. You see that it's actually a double membrane, and inside of it we have an area called the nucleolus. So those are two words that look and sound similar, nucleus and nucleolus. Nucleus is the whole deal. Nucleolus is a region, a dense region inside the nucleus where ribosome components are made. All right, we've got plasma membrane on the outside. We've got mitochondria, and we're going to go over each of these organelles. Mitochondria are the site of cellular respiration, and they are making ATP, generally using glucose. So mitochondria are the site of cellular respiration, and they're converting the energy in glucose to ATP. You may see or hear someone say that the mitochondria are producing energy. No, no, no. We cannot create energy. We can convert the energy in sugar into the energy in ATP, but we do not make energy. Mitochondria do not make energy. You may hear them called the powerhouse of the cell. I prefer you don't use that term. They're the site of cellular respiration, right? And their function is to make ATP. And you'll learn more about cellular respiration coming up. This labyrinth of membranes that's shown in blue is endoplasmic reticulum. Endoplasmic reticulum. This is definitely membranous. So this is a labyrinth of membranes. You see that it's continuous with the nuclear envelope, right? So it's just continuous with the nucleus. And you see all these little dots on it. The little dots represent ribosomes, which is site of protein synthesis. And then you see some that's smooth. This is smooth ER with no ribosomes. If it's rough, it's got the dots. It's got the ribosomes. So we have two kinds of endoplasmic reticulum. They're both membranous. They're both attached to one another and to the nucleus. Rough ER has ribosomes and produces protein. Smooth ER does not have ribosomes and produces lipids. We have another membranous organelle called the Golgi apparatus or Golgi body or Golgi complex. All of those mean the same thing. And so here you see the membranes and they're kind of stacked on one another. It's more like a layer of pancakes there with some blebs coming off. So you see these smaller vesicles coming off of the Golgi apparatus. So vesicles are moving from the ER to the Golgi apparatus and then moving from the Golgi apparatus to the plasma membrane or other parts of the cell. We have the centrosome, and its function is going to be control the microtubules during cell division. This is representing a mitochondria that's been cut open so we can see what's in it, and these are mitochondria just looking at them from the outside. There's our flagellum, which in humans is only found in the sperm cell. All right, let's go forward and we'll get a closer look at these organelles as we go through the chapter. Here is our plasma membrane, eukaryotic plasma membrane. This is going to be very important to understand. We have a lipid bilayer, phospholipid bilayer. So when we say bilayer, that means that we've got two layers, right? And it is made of phospholipids. And these little heads that we're seeing are the phosphate end of those phospholipids. So there's one molecule of phospholipid right there. Here's a protein channel. We've got some other proteins. This is called a peripheral membrane protein because it's only partway through. An integral or integral membrane protein is going to go all the way through. We've got glycolipids that allow for attachment and recognition between cells. There we have a glycoprotein, so that's a protein with carbohydrate attached. We've got cholesterol in this membrane, and this is considered a fluid mosaic. Fluid mosaic. So fluid means that it moves around, right? It's not a stack of bricks. These are molecules that can move around. And mosaic meaning this is made of lots of small things put together. All right, we see the filaments of the cytoskeleton. This is the inside of the cell. This is the outside. So we have an aqueous environment outside and we have an aqueous environment inside. These phosphate heads are hydrophilic. Hydrophilic means they like water. These tails that are in between the inside of the membrane are hydrophobic. Hydrophobic, they don't want to go into water. Remember our salad dressing, right? That these are the oil and they don't want to be in the aqueous environment. All right, so the cytoplasm is the region between the plasma membrane and the nuclear envelope. And that is both the gel-like cytosol and the cytoskeleton and the organelles, right? So cytoplasm is all the stuff between the plasma membrane and the nuclear envelope. The cytosol is the aqueous portion of the cytoplasm. 70 to 80% of the cytoplasm is water, has a semi-solid consistency due to the proteins within it. We can think of it as sort of a loose gel. All right, let's have a closer look at each of these organelles. Nucleus, usually only one per cell. In biology, we have so many exceptions to things. In muscle cells, in skeletal muscle cells, we will have more than one nucleus. But for the most part, let's start with just one nucleus per cell. In fact, in red blood cells, we don't have any nucleus. Usually the largest, and it alone is larger than most prokaryotic cells. Prokaryotic cells do not have a nucleus. Nucleus is only present in eukaryotic cells. Keep those straight. There's often exam questions on prokaryotic and eukaryotic. All right. Again, our nucleus is represented in purple, but it is not purple. The nucleus contains the DNA. The nucleus contains the genetic material. And inside of the nuclear envelope, we call that stuff nucleoplasm. Nucleoplasm is our gel-like stuff inside the nucleus. This squiggly stuff is the chromatin, that's the DNA. We've got our nucleolus, which is a denser area inside the nucleus. So these sound and look similar, but we want to keep them straight. In the nucleolus, the cell is producing ribosome material. There's our endoplasmic reticulum. In this case, they're showing us rough ER. It's rough for the ribosome dots, right? It's the ribosomes that make it look rough. The ribosomes show up in transmission electron microscopy. The openings in the nuclear envelope are called nuclear pores, and we'll learn about that later. The messenger RNA comes out through that nuclear pore. That nuclear envelope is a double membrane, right? It's a double membrane. So it's going to have 4 layers, right? Because a membrane's got 2 layers. Double membrane, we've got 4. We've got these little nuclear pores. Did we cover all of that? All right, know your nucleus. Nuclear envelope. Nuclear envelope. So right now we're just looking at that section. It's a double membrane. It separates the DNA from the cytoplasm, separates transcription from translation. We'll talk about that a little later. Transcription is the production of RNA using a DNA template. So transcription, in the process of transcription, we're making a messenger RNA and we're using DNA as the instructions. In translation, we're using the instructions on the messenger RNA to make protein. And that's going to happen outside the nucleus. All right, so the nuclear pore is an opening in that membrane, connecting the nucleoplasm to the cytoplasm. It's regulating the flow of molecules back and forth, and the large molecules are going to have a hard time getting in, so they're going to require this nuclear localization signal to pass. So there's our inner membrane, our outer membrane. There is our nuclear pore, and we've got a little space in between, and it looks like we got some ribosomes on there too. So that messenger RNA is going to come out through the nuclear pore for that messenger RNA to be translated. Nucleolus is a region inside the nucleus where ribosomes are assembled, and we're going to use RNA and proteins to make those ribosomes. So that's that dense area inside the nucleus. There may be more than one. Okay. And when you're looking at these illustrations, always get oriented, right? You might look at it and say, okay, we're talking about nucleolus. Oops, there it is. No, get oriented on the whole illustration. All right, ribosomes. Ribosomes are the site of protein synthesis. Their function is to make proteins. To synthesize means to make. So they are actually made of two different subunits, and these come apart. There's our large subunit, our small subunit. They come apart, and they are larger in eukaryotes. They do occur in prokaryotes, because remember, they don't have a membrane. So they are made of rRNA. We have several types of RNA, and we keep finding more, right? So rRNA is the RNA that is inside the ribosome. We've also got proteins in it. This is showing us a ribosome that is busy making a protein. Right? So there's our small subunit. Here's our large subunit. This is our messenger RNA carrying the instructions for the protein. Here we've got a transfer RNA, or a tRNA. And here we have one amino acid. And as we stick those amino acids together, we call that a protein, a peptide. So if it's less than 100 amino acids, we call it peptide. If it's longer than that, we call it protein. And so as they're making these proteins during protein synthesis, the ribosomes assemble the amino acids into proteins in a particular sequence. A sequence means an order. So those amino acids are going to come together in a given order based on the messenger RNA. Mitochondria are the site for conversion of stored energy. So that energy is stored in the molecules. They're talking about macromolecules. We're usually thinking of sugar, which is a building block. And they are converting that energy into ATP. The inner membrane is folded, and these folds are called cristae, and the area enclosed is called the mitochondrial matrix. So we've got our outer membrane, and we've got this inner membrane that's folded in on itself, right? And inside that inner membrane we have the matrix. There's a lot more going on here, but this is easily recognizable. The mitochondria is easily recognizable in an illustration. This is showing it cut open, and its function is to form ATP. It is the site of cellular respiration. Peroxisomes and lysosomes are really difficult to distinguish in an unlabeled model or illustration, so I would not ask you to say, oh, this is a peroxisome and this is a lysosome in a given illustration, because they're vesicles that look the same. But as we're talking about them, we can know the difference, right? So peroxisome are these basically a vesicle. We've got a lipid bilayer, we've got a membrane around it. So we've got a single membrane, and it breaks down fatty acids and amino acids. And these peroxisomes may also detoxify poisons. And they just, in an illustration, they're just going to look like a... Let's talk about some important differences between plant and animal cells. Animal cells have centrioles, and that's associated with cell division, and it forms a complex called the centrosome during cell division. So animal cells have a centrosome and lysosomes. Plant cells do not. Plant cells have a cell wall, a chloroplast, and they also have a large central vacuole, and animal cells do not. Plant cells, cell wall, chloroplast, central vacuole. Animal cells, centrioles, lysosomes. No cell wall. All right, here's our generalized animal cell. So when we say generalized, this is like kind of throwing together different things that you might see in animal cells. It is showing us a small vacuole, but not a large central vacuole. Here's a little secretory vesicle, so that's something that's being excreted from the cell. We've already talked about plasma membrane. We've talked about mitochondria. The ribosomes can be on the rough ER. They can also be free out in the cytoplasm. We talked about our nucleus and nucleolus. The chromatin is the genetic material that is inside the nucleus. The Golgi apparatus is a membranous organelle, and here you see them stacked one on the other. This is continuous, right? This is a cross section that looks like they're layered on one another, but this is all connected. And we see these Golgi vesicles being released from the Golgi apparatus, and they are moving towards the plasma membrane, where they can meet with that membrane and release the contents into the extracellular space. There's a lysosome. There's our smooth ER. There's our peroxisome. And the only difference here we really see is in color. We have no way of telling from one unlabeled illustration to another which is which. There's our centrosome that will play a role in cell division. We also have filaments that make up the cytoskeleton. We have an intermediate filament, we've got microtubules and microfilaments that make up the cytoskeleton that can move these organelles and vesicles around the cell. By contrast, in plant cells, we have a cell wall. Plant cells tend to be more angular, right? Kind of squared off, or have angles, not just kind of be loose. So the cell wall maintains the shape of the cell wall. It may be continuous. Two cells can share a cell wall. We still have the plasma membrane, of course. We've got the mitochondria that we've already talked about. We have the chloroplast, which is green. So that's the organelle that has color. These other organelles don't really have color. Chloroplast is the site of photosynthesis. Their function is to make sugar by capturing sunlight energy and fixing carbon. We talk about that in a different class. We talk about that in Bio 3. We won't spend too much time on that in human biology. We still have the cytoskeleton. We've got peroxisomes. We've got the mitochondria. So even though plants are capturing sunlight energy and making sugars, they're still going to need to burn those sugars sometimes. We've got ribosomes, and then again we have our Golgi apparatus, also called the Golgi body, right? Again, we've got our cytoskeleton, and then we can also have our central vacuole, which can play a role in helping that cell to grow, helping the plant to grow during the springtime, during the growing season.